and welcome to
You Cannot Be Serious! What a great name we've got there.
I agree.
Okay, so my name is Kate.
I'm Carly.
I'm Dan.
I was so enthusiastic.
Thank
Dan's
you.
very excited to be here today.
As you may have noticed already, we have a new person here.
Hello.
So Dan, what kind of research do you do?
Well, Kate, I'm in the same lab as you, so I also do research on DNA damage. And I'm just starting, so I'm not doing a lot. I'm doing grunt work at the moment. So fun.
That is okay. You will get up to my level soon.
Okay.
And what level is that, Kate? I must ask. What is that?
Elite scientist, obviously.
We're all elites here.
Okay, so what we will be talking about today is the central dogma. So the central dogma is how DNA goes to RNA goes to protein. So DNA to RNA is called, that process is called transcription. And RNA to protein is called translation. And then at the end of the podcast, we're going to talk about a cool article about how squids will alter their RNA to better survive in
Cold temperatures.
In cold
In
temperatures.
cold temperatures. It's actually
I wish I could do that.
survive in cold temperatures.
Yeah. I don't like the cold.
That's fair. Yeah. That's fair.
So to start out, we are going to talk about DNA to RNA. Carly, can you tell us more about transcription?
Transcription, we are going to break this down in a way that makes sense. So, as Kate said, DNA to RNA is a process known as transcription. In transcription, there are three, I guess you could say, main steps that take place within, of course, your cells. And these are called initiation, elongation, and then termination. I actually had to do a presentation on this, so I don't know if you guys remember that.
I don't.
Okay. As far as initiation goes, this starts when your RNA polymerase bind to a promoter sequence, and then it's kind of recruited here by all kinds of things called transcription factors. So these are kind of like, hey, this is where this should kind of happen. So,
And side note, my favorite thing in transcription is that one of the promoter sequences is called a Tata box.
yes.
We were talking last time about how silly some of the names are, and it makes sense why it's named this because it's TATA for the nucleotide sequence, but still, it is so satisfying to say Tata box.
In the brain, there's a scientific term. It's called blobs. And it helps you with your vision. So, in part of like the little neuron network to help you see, you have little blobs. Scientific term.
The more you know. So, this RNA polymerase is able to split your strand into two. So now you've got two strands. So, elongation is the next process to occur. So here, this is when one of your strands becomes a template, and then one becomes the coding strand.
So, the best way I could think about it is that it kind of goes reverse. So you have your template strand, and then you get your mRNA under it, which is the complementary sequence. And then it becomes the coding strand, but in mRNA. But the difference is that mRNA has U for uracil instead of T for thymine. I almost said thyroid, and that is not correct.
We don't even
I know it was a thigh, but that's how I remember it. So the coding strand is the same as the mRNA strand with just one letter swapped out. But really it goes from the template strand to mRNA.
So, like, if your teacher ever asked you to go from DNA to mRNA, you can just look at the coding strand and just copy that, but make sure to change your T's to U's. But your body uses the template strand actually to make the mRNA.
Yes, so then if you are asked to go from, or I guess like write the sequence from DNA to RNA, you have to remember the base pairs. So DNA is composed of the bases A, T, C, and G, while RNA is A, U, C, and G. So A and U will be little pairs, and then C and G will be pairs. Unless you're a squid, which we'll talk about later.
Love the segue.
It's actually so cool. So we'll talk about that. And I guess in this simplified version of transcription, the final phase is termination. The RNA is kind of going along, the RNA polymerase is going along, and it will encounter a termination sequence, which will basically say, stop! And it stops.
The RNA polymerase falls off of the DNA, and that will cause the mRNA to stop being created. And since the RNA polymerase has fallen off of the DNA, the mRNA will then be finished. And additionally, when mRNA is created, it also has to be fixed up and made a little bit pretty in kind of a sense. So it needs a 5' cap and a poly A tail. So these things are just little things we add to the ends of the mRNA to prevent degradation, so breaking down of the mRNA. So it's the thought of adding something to the ends of the important mRNA so that if you break down the ends, you're breaking down unimportant information.
So at this point, right, transcription was just terminated, that RNA polymerase is gone, it fell off, so the process has been stopped by that like stop sequence, basically, that's at the end or wherever it is in the RNA at this point. So the next portion will be RNA actually being turned into a protein, and this is through translation. I don't know who wants to talk about that, but go for it.
I'll talk about it.
Cool.
Okay. So translation actually occurs via ribosomes and tRNA. So the mRNA has come out of the nucleus, and so we are now in the cytoplasm. So the ribosomes reside in the rough ER. So if you think about, if you ever made like that annoying model of the cell, you know, I had to do that.
We've all had to make. Did you not have to make a model?
What do you mean?
You know, like you had like a styrofoam ball, and then you had to like cut. No, you never had to do that.
Oh, see, mine.
No.
I had like a piece of cardboard like this big, and we had to model it like out of clay on the board.
Yeah, I used clay to make all the
okay.
organelle, but like I literally took a ball of styrofoam and like cut it so that it looked like a cell.
Oh, that's
like
kinda
cut
cool.
into it. Yeah.
I did not have this fun experience that y'all had. I did not make anything out of clay.
Well, okay, Dan didn't have fun education.
No.
But if you like think back or think back to pictures in your biology textbook, you'll remember that like the rough ER is the one that looks like a kind of a ribbon, and has like almost like little dots all over it.
Mm.
And those are ribosomes. So ribosomes are where we make protein. So the mRNA has now entered the cytoplasm, and the mRNA is going to meet up with a ribosome, and it's going to also meet up with a tRNA. That the first tRNA is going to be attached to a methionine. So this is because, this is important, every protein starts with the start codon AUG. So that is just the start codon is AUG, and AUG connects with methionine. So breaking down codons and amino acids. So amino acids make up proteins. If we think back to like the beginning of like biology class, like that first lecture your teacher gives, and it's like macromolecules, and you're like still sitting in class and you're like, what is this? Your teacher probably told you amino acids make up proteins. So tRNA has a little anticodon, and that matches to an amino acid. And then that anticodon matches to a codon in the mRNA. And a codon is three nucleotides. So the three nucleotides A, U, G. And the tRNA will recognize AUG and bring over methionine. And it will land in the ribosome in the middle spot. So a ribosome has 3 spots. So it will land in this middle spot. So let's say the next codon in the mRNA sequence is AGG. We want to know what tRNA is going to come next. So we would look up the codon chart. You just went on Google
There is a chart.
There is a chart. If you just went on Google and Googled codon chart, 100 would come up.
Oh yeah.
And you just find your favorite, the one that makes sense to you. And we would look at it, figure it out, and we would find that AGG matches to arginine. So the tRNA that is connected to arginine comes over and lands in the first spot. Then the ribosome shifts and moves the tRNA with methionine over to the third spot, and the tRNA with arginine to the middle spot. A bond is formed between methionine and arginine, beginning the polypeptide chain. And the bond between the tRNA and the methionine is broken. Then the tRNA that now does not have any amino acid, but that was carrying the methionine, now leaves the ribosome. So now there is just the tRNA with the arginine and the methionine is in the middle spot. So now the process can start all over.
It's kind of a lot.
It is a lot, and if you listened all the way through to right now, I'm very proud of you.
Another thing about the codon charts and proteins in general is that Kate was saying methionine. You also see it as met.
Mm-hmm.
You also see it as, well, you normally kind of just see it as start, but basically they all have a little 3-letter code instead of the full name, and also a 1-letter code to shorten it. So you pick your favorite codon chart, make sure it has what you're looking for. Because sometimes they won't show the full name. Sometimes they will.
Mm-hmm.
Sometimes they'll just show the 1-letter code.
Mm-hmm.
And that can be confusing because when there are 5 different things that start with a T, but only one of them can be labeled as T, then they start moving to other letters, and it's just, yeah, you just got to be careful when you're doing that, because it's not going to be exactly what you think every single time. Are we ready for cephalopods?
I'm ready for cephalopods.
Okay, so we just kind of highlighted, going from DNA to RNA through transcription, and then from RNA to a protein. And now we get to talk about something really cool that happens not just in cephalopods, but for the purposes of this, we're going to be talking about cephalopods because these articles were like very interesting to read.
Yeah.
So they go through this process called RNA editing, and there's an even more specific name when it gets into the specific base that it is editing. But we'll get into that later.
So So
I can talk about it. I don't care.
no. Okay. So
No.
no, you may not talk. Okay.
I didn't read this paper. This is new to me.
Yes.
Oh, it's really it's really cool. Actually, I was talking about it with one of my lab mates earlier too.
See, I just like eating cephalopods.
I thought you were just gonna say eating, and I was like, yeah,
Yes.
yeah, same.
Okay. But as previously mentioned, the RNA is made up of 4 building blocks: adenine, uracil, cytosine, and guanine. So the cephalopods are able to change their RNA through RNA editing by switching out adenines with another nucleotide called inosine. So
I have no clue how you pronounce that.
I don't know
But it's
either.
labeled with an I.
Yes.
So we're just gonna call it
Inosine.
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 I have never seen it. I have never read it. I
Well,
have.
if we go if it's like guanine,
Yeah.
inosine, or cytosine,
I think so. It has to be.
I think you can go either way.
Yeah, it is. A to I editing via the enzyme ADR2. So basically, what this does is it takes off. I forgot what it takes off, but it changes
Nitrogen.
nitrogen
And it takes off, I think, 1 nitrogen and 2 hydrogens.
off of the nucleotide, and good job. And so the body then reads the I as a G. So if we think back to how transcription, translation, everything, specifically translation, happens, every time there's an I, it's going to read it as a G. So that's going to change how the codons are read. So if we think back to the arginine, instead of AGG, it's going to be GGG. And that's going to change what amino acid is brought over. So
I love this protein. I'm sorry.
Then talk about it.
Kinesin is so cute.
Why is it
I
cute?
literally had to make a note because I remember in high school. I'm so sorry if I
No,
cut you off. It was like the next thing to talk about anyway. I just think kinesin is so cute. You don't think? Have you seen the videos?
yes, I have. I never thought
yeah.
cute when I saw
Oh my
it.
god, it's so cute. Are you kidding?
Its little legs.
His little legs walking
But I mean, technically, like anything has those little legs.
But kinesin is cute because it's so tiny.
Okay, so what she's talking about
Kinesin.
Wait, how does that relate to the squids, Carter?
You'll listen. So kinesin is this motor protein in your cells that kind of walks along your microtubules, which are within your cell, help with structure, integrity, all the fun jazz. And it has a microtubule binding domain, and this is able to change, so it's able to literally walk along your microtubules and carry all kinds of stuff. I've seen it can carry organelles or other molecules. So it can carry all kinds of things throughout your cell just by walking on your microtubules. So it can literally get anywhere. But
So-
with the squids,
Yeah.
squids,
Cephalopods.
obviously this happened in a lab. So they initially had little squid hatchlings, and they put some of them in 6 degrees Celsius, I believe, and then they put some other ones at a more, I guess, like normal temperature. This was 20 degrees Celsius, and they observed that this editing that Kate was previously talking about is also happening in kinesin. So they saw that between the different water temperatures, there's something different going on with kinesin itself. So they then were able to, I think they did some molecular editing themselves on kinesin, and they observed this unedited kinesin as well as edited kinesin to kind of see and explain what was going on within the squid.
So what happened was that they found that when the squid was in cold temperatures, the kinesin would slow down and fall off of the microtubules. But in response to that, the squid would edit its RNA, its mRNA, and this would fix the kinesin. It would hold on better to the microtubules and move faster and longer distances.
Cool.
So it helped them like stay mobile in cold temperatures, basically.
Yes.
Yeah.
Interesting.
Right?
Yes. And so this only happened when they were exposed to the cold
Yeah,
temperatures.
so it doesn't happen in the 20 degrees Celsius, but it did in the 6.
Yes. And so why were they doing this? Like why? So if we think about like evolution and changing so there can be mutations that occur and they're passed on to offspring, and it can make the generations and the organisms more fit to their environment. So why wasn't that occurring? It's because the temperature was fluctuating. It wouldn't have been beneficial to change their DNA.
Right, because it's permanent.
Yeah, the genes are permanent. Any change to their gene was going to be permanent. But if you just turn on and off an editing to the RNA, then you can change that depending on what temperatures are in.
So did they only see that A to I?
Yes.
They didn't see it. They didn't see any other change. They only saw that one
At least
specific change.
in the article, 'cause we looked at a few. That was the main one that they mentioned. Now this might be something that needs to be looked at further because, if it implicates cephalopods, who knows what else it's affecting as well. But yeah, they mentioned mostly the A to I, and it has a specific name, RNA. I think it's called recoding.
Yes.
I don't know. I'd be interested to look even further at research that's going on with all the time that I have in the day.
We're very busy. So it's just very interesting to see how different animals are changing their RNA expression to their environment, and how it's not even an adaptation because that's evolution and changing your genome. It's changing RNA expression to better fit your environment and the changes in your environment.
Well, okay, to push back a little bit on that
Yeah.
statement, I think it is a testament to evolution because they have in their genome the specific gene that allows them to RNA recode. That's not something, as far as we know, it's not something that everything can do.
Yeah, I
Right?
agree with that.
It's just interesting to see how, like, if you think about bears and hibernation and stuff like that, it's both like two different responses to the cold almost.
Right.
Okay. So let us know if you have any questions about this, any comments, your thoughts. So what have we learned today? We have learned the central dogma. We have learned about transcription and translation. And we have learned about really cool squids and how they can edit their RNA and how that relates back to transcription and translation. So again, thanks for tuning in, and we hope you enjoyed it.
I'm never coming back.